Dynamic brain and neuroplasticity. Involvement of Homo and Heterosynaptic plasticity. We know that plasticity refers to the ability of organisms or cells to adjust their phenotype in response to alterations in their environment. In neuroplasticity or brain plasticity, the brain synapses are modified by environmental thinking, emotion, behavioral, and neural alterations throughout the brain development. Two kinds of plasticity, homosynaptic plasticity and heterosynaptic plasticity are induced in synapses. Homosynaptic plasticity is induced in synapses that were directly activated in the neuron during the development that needs the presynaptic activation. It is input specific or associative, has the associativity because it associates the presynaptic and postsynaptic neuron activity. On the other hand, heterosynaptic plasticity can be produced at synapses that were not active during the induction. Any synapse at the cell can be subject to this kind of plasticity. But totally, both of them are required for normal action and balance the synapses. Let me to explain you one of our studies on layer 6 of the visual cortex and one type of a specified type of the cells, corticogeniculate neurons, CG cells. CG cells can receive two important input from LGN, lateral geniculate nucleus, and also from upper layer, especially layer 2 and 3. This layer can affect both of them, thalamocortical and corticothalamic lobes, and which mitigate the information that ultimately ascends to the higher cortical layers. In this study, we use we get a great labeling in the CG cells. For this aim, we use alexaflor and we inject, this is LGN of the thalamus, and this return in the layer 6, you see. Here is visual cortex, layer 1, 2, 3, 4, 5, and finally 6. Other fluorescence microscope, you can see the cells. These are CG cells that retrogradely are red, and they are these green ones are GABAergic neurons. So we can completely recognize them. I mean, CG cells and GABAergic cells. In this study, we used, as I told, we had two input from LGN that can reach here from white matter, and from upper layer, especially layer 2, 3. If we use TBS to upper layer, so we can induce homo-LTP in this synapse. On the other hand, in other synapse, hetero-LTP will be induced. Then we search about their mechanisms. This homo-LTP from upper layer was angular 5 dependence and was post-synaptic. It was due to postsynaptic mechanism. And this hetero-LTD was presynaptic and dependent on endocannabinoids from induction of homo-LTP in other cells. On the other hand, if we use TBS to white matter, we could induce LTP in this synapse that was NMDA receptor dependent, and in other pathway in upper layer, we could induce hetero-LTP. So this first one.
Arami, you are going over a little bit of your time. Could you please, Arami, could you please, wrap it up?
This is a top down and this is bottom up. So in this study, we could compare these two flow, bottom up and top down flow, and so we can explain the gain control function of seizure cells in layer 6 that is regulated by reciprocal long-term plasticity of synapses converging from the upward and downward pathways. Thank you.
Thank you. And